This figure is showing an example of one of Mendel's crosses. And the first thing Mendel established were what he called true breeding plants for each of the two phenotypes for the particular traits. So in this example, we're looking at tall plants versus dwarf or short plants. And so by controlling the matings, he produced true breeding, or a more appropriate term would be homozygous, meaning they had two of the same allele types present within them. So these true breeding plants for the opposite forms of the phenotypes were said to be his P generation, or his parental generation. So what he did was he crossed these plants, and in looking at these two plants, he had no idea which trait was dominant. The only way you can figure that out is by putting the alleles together. So when this plant produces gametes, the gametes will have one of its alleles, and since it's homozygous, there's only one type of gamete produced. This one will produce one type of gamete with respect to allelic content. So when the heterozygote, or the individual containing two different alleles, one from this form and one from this form, were put together, all Mendel saw in the F1, or the first generation, were tall plants. So on the basis of that, he presumed that tall was dominant to short. Now, what he did to produce his second generation of progeny was he intercrossed his F1s. So he did an intermating. And in doing this intermating, he found that 3/4 of the offspring were tall and 1/4 of the offspring were short in terms of phenotype. And that was his characteristic phenotypic ratio. In his F1, he saw all dominant form. In his F2, he saw a 3-to-1 form. Now, if we apply allele symbolism to this, it will make sense. And you're going to need to get a piece of scratch paper and follow this with me. If tall is dominant, then we're going to designate this allele as uppercase T. Since it's true breeding, the genotype for this plant would be homozygous dominant. In other words, uppercase T, uppercase T. The genotype for the true breeding short would therefore be homozygous for the recessive allele, or in other words, little t, little t. Now, when these two plants produce gametes, they will only produce one gamete type with respect to allelic content. The tall will produce all gametes with a dominant allele. The short will produce all gametes with a recessive allele. So if you put a dominant allele together with a recessive allele, we get a heterozygote, or in other words, the genotype would be dominant T, recessive T. Since tall is dominant to short, all of the F1 plants will be tall. Now, if we intercross the heterozygotes, I'll have a big T little t times a big T little t. So what that means is each of these plants produces two types of gametes in equal frequency. Half of the gametes would have a dominant T, half the gametes would have a recessive T. And this is where some of you may have learned the concept of a Punnett square. And what a Punnett square does is represents outcomes of fertilization. So what I encourage you to do is to, and you'll have to imagine this with me, draw a square, and inside that square draw a vertical line, and then a horizontal line. So you'll split it up into 4 cubes. On the outside, put a dominant T, and in the next box, on the outside, put a dominant T, and by the next row, put a recessive T. Up top, on the first column outside the box, put a dominant T, and then a recessive T. Those represent the gamete types produced. So then you represent the outcomes of fertilization by putting the alleles with each other. In other words, if my very first box will have a dominant T on the outside to the left, it'll have a dominant T above it, and if I put those two dominants together, I get homozygous dominant, big T, big T. In the two boxes on the diagonal, I will have a heterozygote, a big T, little t. And in the last box on the lower right-hand corner, I would have a homozygous recessive, little t, little t. Now, those would be my genotypic ratios. I would have one homozygous dominant to two heterozygotes to one homozygous recessive. In terms of phenotypes, however, because of complete dominance, the homozygous dominant, the two heterozygotes will all show the same phenotype. Therefore, they will all be tall. So even though I have different genotypes, they all have the same phenotype. So 3/4, 3 of the 4 squares, would have a genotype that resulted in the tall phenotype, whereas only 1 of 4 squares, or 1/4, would have the homozygous recessive genotype, resulting in the recessive phenotype, or short. So what we've done is we've followed a trait using allelic symbols from the P generation to the F1, or the first generation of progeny, and then from the F1 to the second generation progeny, or F2. And those of you that take the lab class, Biology 163, you will do crosses of this nature, except instead of using pea plants, we'll use Drosophila melanogaster, or fruit flies. And so I think it's a really interesting experiment to actually look at inheritance of traits. Instead of just looking at a figure like this, you actually see the biology of the organism, you count the progeny, you determine their phenotypes, and I think it gives you a much better appreciation. Now, this is called a monohybrid cross because it's only dealing with one trait. So this is a monohybrid cross for the trait of tallness of the plants, and this also illustrates Mendel's first law, which is the law of segregation. In other words, during meiosis, the genotype splits and is packaged into gametes. So big T splits into all big T gametes, little t split into all little t gametes, and we get one genotype for the F1, heterozygous. In production of the gametes for the second generation of progeny, the heterozygous genotype splits. Half of the gametes are going to be dominant, half would be recessive. So again, a restatement of Mendel's law of segregation is during meiosis, alleles of a gene separate and they're packaged into gametes.